Okay, TCA cycle. Now we're still, just to remind you, we're still in this phase of our fed fasting graph here. We're still here at number 2. So we're at the peak. We still have high blood glucose levels. We have insulin signaling in the fed state. We have GLUT4 on the surface of muscle cells. It's taking in glucose. We're generating energy. So we're still in the energy generating phase. Now, TCA. The TCA cycle. So most of you probably have learned biochemistry and the fact that glycolysis is first. That's what you learn first. And then from there you go into TCA. But this is where you need to start changing your thinking, because in a medical sense, the TCA cycle is the center of the universe. All these other pathways feed into the TCA cycle. Disruptions in the TCA cycle can cause a wide array of diseases because substrates come into the TCA cycle and then other molecules come out. So the TCA cycle is the center of fatty acid synthesis. It's the center of gluconeogenesis. It's the center of amino acid oxidation and amino acid synthesis. So that's why I say everything comes in and out of the TCA cycle. So it's the center of the universe. So here it is. We're reactions, right? Okay. So what I'm going to do is I'm going to break this down into the key regulated elements that are going to control TCA activity. I have not seen any questions. I've taken them out of my database to ask you, what follows alpha-ketoglutarate. That's just really silly. The NBA database, I have not seen, yes, just, I want everybody to do a slam dunk. Ooh, that was good. The NBA database, I have not seen them ask you specifically, what substrate and product follow each other and what enzyme is done in what place. They will ask you questions like, if this enzyme is defective, what is the consequence? So how do you interpret this? Well, look at the enzyme that's defective, back up in the cycle, think about the substrate, and again, as those substrates start to build because you've got a block, what's the escape route for that substrate? Okay? Same thing that we've seen with skeletal muscle. If you've got a defect in pyruvate dehydrogenase, pyruvate builds up and it's got to have an escape route. Because if it doesn't have an escape route, that's going to cause cell death because that's an acid. So its escape route is lactate. Lactate goes into the blood, metabolic acidosis. Okay? So that's kind of the way a lot of these questions are looking at. So what do I mean about regulation? So let's focus on this cycle and let's look at the very first reaction right here. This is a condensation reaction of acetyl-CoA and citrate. Now, I'm focusing on this very first reaction because the acetyl-CoA is going to condense with oxaloacetate. Oxaloacetate is a 4-carbon molecule. Acetyl-CoA is a 2-carbon molecule. This is where the math gets really easy. 4 plus 2 is 6. So citrate is a 6-carbon molecule. Now, what's important about this reaction is that this reaction is irreversible. Now, if you think back on chemistry, there's not a lot of reactions that are irreversible. And this is another change in thinking. There are certain biochemical reactions in the body that are irreversible because of thermodynamics. What does that mean? This acetyl-CoA This bond right here between the acetate and the coenzyme A is a high energy bond. So this reaction, we are condensing these two molecules together and we are releasing coenzyme A. Now for those of you that still remember your thermodynamics, this is a very large free energy change. Remember Gibbs free energy? If it's a big negative, that means it's a big forward push and it's irreversible in the body. So that means because of this, the reverse reaction would be creating a high energy bond. So you have to recouple the coenzyme A with the acetate to make acetyl-CoA. We cannot do that in this reaction. So that makes this reaction one way and irreversible. So once we tap acetyl-CoA and we put it into the TCA cycle, it's a done deal. You have to go through the cycle. Now, at this point, the citrate comes around, forms isocitrate, blah, blah, blah. That's easy. This enzyme is reversible. This one here is going to form alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. So again, dehydrogenase, this is a redox reaction. So in this reaction, we are generating NADH. So we're extracting electrons. Now, because we have NADH being generated, this situation is going to be the same as PDH. This NADH can inhibit isocitrate dehydrogenase. Whoops, sorry, minus. It can inhibit that enzyme. Now, in skeletal muscle, the purpose of this is the same purpose as regulating PDH. You want to slow everything down so that your NADH levels have a chance to catch up, because this return back to NAD is the electron transport chain. So in skeletal muscle, again, it's very easy. You just want to kind of get off the throttle a little bit and slow the process down. Yes?
Is this a second site for regulation, or is this just a buildup of the product that's causing this?
It's a product inhibition. You're right, right. So it's actually a lack of NAD, the substrate, and a buildup of NADH. So the NADH kind of sticks in the active site of the enzyme too long. Right. So yes, that's a good point. This is not a kinase or phosphatase in regulation here. So now in the liver, this happens also. But in the liver, again, we have our escape route. So this reaction is reversible from isocitrate back to citrate. And now in the liver, citrate can be used to form fatty acids. So when we have a lot of glucose coming through the liver, we have oxidation going on. We have acetyl-CoA condensing in the liver mitochondria to make citrate. The liver is going to use this to generate energy for itself, but if its energy charge goes up with NADH, it can now shift into fatty acids. And so this arrow right here, we're going to go into this in more detail tomorrow, I think. Yeah, tomorrow. But this is probably going to be your number one challenge as a physician. Because excess glucose, we oxidize it for energy, we can store it as glycogen, and when everything is filled and everybody's happy, this is our escape route. The liver's going to make fatty acids. Now, under normal circumstances, making fatty acids is good because we're storing our extra calories. But go back to what Dr. Alcorn was talking about, fatty liver disease. This is all occurring in the liver. We'll talk about how the liver handles fatty acids on Friday, but if you overload the system, now you get steatosis, fatty liver disease. Okay? So that's a little bit of a preview, but that's the regulation, and this is only in the liver, no place else. Okay, so now, let's say that our block is relieved, our NADH levels go down because we are reforming our NAD by electron transport activity. So the alpha-ketoglutarate will form, if I can spell, succinyl-CoA. Now, this reaction here is also a decarboxylation reaction back here. Alpha-ketoglutarate is a 5-carbon molecule. The enzyme that does this reaction is alpha-ketoglutarate dehydrogenase. So this is another redox reaction. We're going to generate more NAD, but this is also a decarboxylation reaction. So now we're getting down to 4 carbons. So the importance of these decarboxylation reactions is that we are finishing up and using our acetyl-CoA, the 2 carbons here, we're getting rid of them as waste products, as CO2. So the purpose of our red blood cells is to transport oxygen to the tissues for electron transport activity, and it's to return CO2 to the lungs, and that CO2 is coming from primarily the TCA cycle. Okay, now, I'll make another point about alpha-ketoglutarate dehydrogenase. This enzyme actually functions in a very, very similar, almost identical manner to PDH. And therefore, it requires a specific vitamin for activity. And that is B1, thiamine. We'll go over these vitamins again to reinforce this later, but this is also a vitamin B1 requiring enzyme. So again, when you start seeing questions that involve vitamin B1 deficiency in a patient, you know automatically that that is going to affect their energy producing capacity, because B1 is required for PDH and it's required for alpha-ketoglutarate dehydrogenase. Without it, PDH is slowed, TCA cycle slowed, energy production goes down. Okay, the succinyl-CoA, we're going to turn this into succinate. This is our point where we're actually going to use this to make GTP. And then our next reaction is going to make fumarate. And this enzyme is very special. Succinate dehydrogenase. So we're going to look at this enzyme in a few minutes also, but this enzyme is membrane bound. And it's located, embedded in the inner mitochondrial membrane. So all the other enzymes of the TCA cycle are soluble in the matrix of the mitochondria. This enzyme is bound, and it's bound for a specific reason. This is a redox reaction, but in this reaction, we are not generating NADH, but we are generating FADH2. So it's just another energy carrier for this. We'll come back and see the significance of that in a few minutes. Okay, so then we've got another reaction here, malate, the fumarase enzyme. There's another very, very rare disease. Fumarase. Does some of you know about the FLDS community on the border of Arizona and Utah, the polygamy sect? What? Oh, you didn't know about this? Yeah. It's on the border, Colorado City and Hilldale. It's a small community. They broke off like 150 years ago from the Mormon Church. And the reason they broke off is because back in the 1800s, the Mormon Church said that polygamy was wrong and they banned it. But this group still believed in Joseph Smith's tenets, and as they formed their own group, and they believe in polygamy. They believe to the point where you have to have multiple wives to get into heaven. And some of them have like 35 wives. I have one. I don't want any more. One is enough. But you can imagine they've been in this isolated community for 100, 150 years. So there's a lot of, what's the word, consanguinity? Did I pronounce it right? That's a tough one. Close enough. So there's a lot of, scientists, we call it inbreeding, right? So there's a lot of, so if you ever see these like Barlows, you'll see a lot of Barlows in the community. So there's a lot of inbreeding going on. So what does inbreeding do? It leads to mutations, genetic errors. So they have a huge frequency of fumarase mutations. So they have this deficiency in fumarase, so obviously their energy producing abilities are severely compromised. And one of the first things that this manifests into, as you can imagine, is mental development problems. Neural development problems. So these individuals have horrible, horrible neurodevelopment because the nerves and all that development needs a lot of energy. And this is actually getting to be quite a medical problem in this community. How do you solve it? I have no idea. You know, it's a closed community. So the government's in there right now trying to modify the community to see what they can do, but good luck with that. Okay. So that's a little side. So that is considered an inborn error of metabolism. Now, I don't recall that ever being on step 1 exams or anything like that because it's extremely rare in the regular population, but I think it can be up to like 1 to 2% in this community. A lot of carriers of that. So the last step is converting malate back into oxaloacetate. And this is why it was important to get rid of our CO2, because we have to get rid of these extra carbons so that we can return back to our 4-carbon starting point so we can condense another acetyl-CoA and go around the circle. This enzyme is a big one. We're going to look at this quite a bit. It's called malate dehydrogenase. Again, dehydrogenase. So this is a redox reaction producing more NADH. And this is the last NADH we're going to produce in the TCA cycle. So we've extracted all of the electrons. We've oxidized glucose at this point as far as we possibly can go. We've extracted everything we can get out of it. Now, what's important about this reaction is the reversibility of it. So dehydrogenase reactions, these reactions here for the isocitrate and alpha-ketoglutarate reactions are typically considered non-reversible. And these are non-reversible because we're generating energy in the form of NADH. To go backwards from, say, succinyl-CoA to alpha-ketoglutarate, we have to create energy. So again, thermodynamically, these have a large negative free energy change. So they're irreversible in the body. This particular one is reversible, but it's reversible only under certain conditions in the liver. This is not reversible in skeletal muscle because skeletal muscle will not have these conditions. We'll go into the details of what causes this reversibility next week. But what's important is that in the liver, malate can be used as a substrate to form glucose. And this is gluconeogenesis. Now, very much like I said with fatty acid synthesis, the synthesis of glucose is going to require a very, very large metabolic shift in regulation, because fatty acid synthesis from citrate occurs only in the fed state. Glucose production and gluconeogenesis only occurs in the fasting state. So that means we need to tell the hepatocyte when we're in a fed state, when we're in a fasting state, so these metabolic changes can occur. And what are the two hormones that tell all the organs that are responsive? Insulin and glucagon. So that's where we're going to start taking all of these metabolic pathways and we're going to overlay another level of regulation. This regulation we're talking about here with NADH, acetyl-CoA and all that, those are intracellular regulations for monitoring intracellular energy levels. Outside the cell are going to be the hormonal controls. Good so far? Okay, there's the TCA cycle. So now, we've got a boatload of NADH. So what do we do with NADH? We see if we don't return NADH to NAD, then everything stops. So now let's look at the electron transport chain. So all of this is going to take place in the inner mitochondrial membrane. And remember, structurally, the inner mitochondrial membrane is the same as those Christi structures. Orientation is really important. So our TCA cycle is going on in the matrix. And this is generating NADH. Malate can be used responsive insulin and glucagon. Cycle. So now let's look at the electron transport chain. So all of this is going to take place in the inner mitochondrial membrane. And remember, structurally, the inner mitochondrial membrane is the same as those Christi structures. I learned that, this is how old I am, I learned that, I could not remember that name, Christi structures in there. So I'm old enough that Christy Brinkley was, like a hottie, super hottie model, 20s. And so that's how I remembered it. I call it the Christy Brinkley structure. So, yeah, that's when she was married to Billy Joel. Anyway, so the electron transport chain. So if this is the matrix side of the mitochondria, this is the inner mitochondrial space, and up here is the outer mitochondrial membrane. Orientation is really important. So our TCA cycle is going on in the matrix, and this is generating NADH. Now the first enzyme in this chain, it's a chain of a series of enzymes, the first enzyme is a transmembrane enzyme, and what this enzyme is called is complex 1. It's also called NADH dehydrogenase, because this is going to perform a redox reaction. So the NADH is going to be converted back to NAD, and what's coming off of here are the electrons. 2 electrons. Now, electrons are extremely reactive and dangerous. So when we talk about oxidative damage in a cell, and we're going to talk about this especially in terms of diabetes, because one of the big factors of diabetes is oxidative damage in a cell. It's largely due to the electrons getting loose. So what we have to do is we have to put these electrons on an intermediate carrier so they're bound and held. And these are called iron-sulfur clusters. So in this case, the iron is different than it is in the iron in hemoglobin. This iron is going from a ferric state to a ferrous state. So the iron plus 2 iron is what is bound to the heme structure in hemoglobin. And this readily binds oxygen. But we don't want this in the mitochondria. We want Fe3 plus. And so here we're going to be able to reduce this into 2 plus by adding electrons to it. So it's the iron in these iron-sulfur clusters that is holding the electrons. Okay? Now, the next step is to transfer this to CoQ. We're transferring the electrons. So the electrons, we will return the iron back to its ferric state, and the electrons are going to be transferred to this small little molecule called CoQ. CoQ is a lipophilic molecule, and it's soluble and embedded in that inner mitochondrial membrane. Now, whenever you move electrons, this is physics. So electrons hold energy. If you move electrons from one physical state to another, you can take advantage of declining voltage potential. So that means you are releasing energy, moving electrons down its electron gradient. So remember I mentioned about the first principle of thermodynamics, first law, that you cannot lose or create energy. So what we're going to do is take these electrons moving down, and this is going to give off energy. What we're going to do with that energy is we are going to pump protons from the matrix into the inner mitochondrial space. So we're converting potential energy of these electrons into kinetic energy to move this proton into the inner mitochondrial space. We're going to keep generating this down the way, this proton gradient. Now, CoQ10, I'm sure all of you will have patients tell you that I saw in this commercial on TV that if I take an enzyme, they call it an enzyme supplement called CoQ10 that you can buy in any store, that it'll give me more energy. You might want to take this before summatives. No, I'm just kidding. So this is where it's important that you be able to counsel your patient when they ask you, how is this going to create more energy? This CoQ10 they're taking is the same as the CoQ supplement that's causing these electrons to move down this chain. So how would CoQ10 supplement increase a person's energy?
Just to shuttle electrons better.
Just to shuttle electrons better, yeah. You're providing more shuttle here to transfer electrons from complex 1 to complex 2. So if we're pushing more electrons, that means that we can harness more energy to make more ATP. So now we are going to shift these electrons from CoQ into complex 2. So complex 2 is going to have these iron-sulfur clusters in order to bind and hold the electrons. There's also cytochromes. Small little proteins, part of this complex, that are going to have heme structures. And of course the heme will also have iron bound to it. So once we pass complex 1, we now get into another type of electron acquisition. So there's 2 types of holding electrons. This is this is complex 3. Got ahead of myself. So you're moving electrons from complex 1 to CoQ, CoQ to complex 3. Where's complex 2?
TCA.
TCA. So I mentioned about this succinate dehydrogenase enzyme, that it's membrane bound and it has converting electrons into FADH2. That is located right here. So this is complex 2, and it's also called succinate dehydrogenase. Now, once we have FADH2, we have to return to FAD, or we can't keep our TCA cycle going. So these electrons here are actually placed into CoQ. By moving these electrons into CoQ, we return to the FAD oxidized state, and now we can continue the TCA cycle. And importantly, we can take these electrons and put them into the electron transport chain. So once these transfer to complex 3, complex 3 is now going to transfer these to a small protein that's called cytochrome C. And again, we're moving down our electrical potential with these electrons, and so we are going to capture that energy in order to pump protons into that intermitochondrial space. So we're acidifying this space. Now, once cytochrome C has its electrons, it's going to transfer this to complex. Since I jumped from 1 to 3, you don't know where I'm going with this one. Complex 4. This also, again, has these iron-sulfur clusters and also cytochromes with heme molecules. And this is the end of the electron transport chain. So the other name for this is cytochrome oxidase. Now, once we have electrons bound to complex 4, we have to unload these electrons so that the chain can keep flowing electrons down. And so what we're going to do here is we're going to transfer these electrons to oxygen to make water. So this is where oxygen is our final electron acceptor. And it's this reaction here is why we breathe. So when we talk about delivering oxygen to tissues, we're primarily talking about oxygen reaching complex 4. So remember how some tissues handle this. If you have plenty of oxygen available in your tissue, you have a nice flow of electrons through your electron transport chain. If you have oxygen lack of oxygen, deprivation. If you have oxygen deprivation, what's going to happen? Stops. The electron transport chain will continue until you become a fully reduced state, where electrons now will occupy each one of these locations. But you can't unload the electrons from complex 4, and so the whole chain stops. What happens to your ATP production? Goes down. What organ do we have that needs oxygen so much that it has its own separate oxygen stores?
Brain?
No. What does stroke do? Kills. So stroke causes ischemia, lack of oxygen to the brain. How fast does the brain shut down in a stroke?
3 minutes.
Yeah, a few minutes. So it doesn't, so that means that if you deprive oxygen flow to the brain, ATP production will slow so fast and use so fast that that brain's going to shut down. Those neurons will shut down. Hemoglobin, what's the other globin? Myoglobin. Why do skeletal muscle cells have so much myoglobin in them? Is it good to be oxygen deprived in your skeletal muscle? No. Think about the 500-pound mountain lion. You want to make sure that your skeletal muscles always have a good reserve of oxygen. And so you can imagine that, so what you've learned, remember your muscle oxygen binding curves? So here's oxygen released, here's your partial pressure of oxygen, here's hemoglobin. So this is the affinity of hemoglobin for oxygen. Remember, myoglobin is like this. Myoglobin is shifted so far to the left on this curve because it has to have a very high affinity for oxygen. So as the hemoglobin is moving through the muscle tissue, you want oxygen transport from hemoglobin to myoglobin. You definitely don't want it the other way. So this directionality is solely due to the myoglobin affinity for oxygen. You can imagine the oxygen affinity for complex 4, is that going to be higher or lower than myoglobin? So if it's lower than myoglobin, that means the oxygen's not going to want to leave myoglobin to perform this reaction. So this reaction is going to have a higher affinity for oxygen than myoglobin. So now what you have is a system to transport oxygen from hemoglobin, red blood cells, into skeletal muscle tissue. It binds as a reservoir to myoglobin, and then it can transfer from myoglobin to complex 4 to perform this reaction to keep the electron transport chain going. Okay. This complex is called the ATP synthase complex. Now, what we've done is we've taken our electrical potential of our electron movement, we've moved protons to create another potential gradient, and now we're reducing this potential gradient and turning this into kinetic energy. So by moving this flux of protons through this stalk, we are going to spin this head. And spinning this head is going to do this reaction. So the kinetic energy of spinning the head is going to create enough energy to couple that inorganic phosphate onto ADP to make ATP. Now, something really important clinically to know about this electron transport chain are chemicals. And these chemicals we're considering are poisons. So poison, so what do I mean by a poison? Well, there's one classic one, rotenone. Rotenone is an insecticide. It's supposed to kill insects. It does. It kills fish, too. So this is one of those insecticides, and there's others that are similar like this. But what rotenone does is it binds to complex 1 and inhibits its activity. Inhibits its activity by blocking the NADH to NAD conversion and electron movement into complex 1. So if somebody ingests rotenone, what type of symptoms would you think they're going to experience? If you stop your electron transport chain. I'm hearing muscle cramps, I'm hearing death. Yeah, it's actually in that spectrum. Because energy is required for everything, right? Breathing, requires energy. Brain function requires energy. All of these different activities that we do to sustain us require ATP. And so if you block your electron transport chain, all your organs, every one of them is going to rely on what process to generate energy? Anaerobic glycolysis, yeah. Is that sufficient to operate this machine? No. These things are incredibly fast acting. And so that's why these things kill so quickly. What's the other thing we just talked about that inhibits the electron transport chain? Cyanide, yeah. That's our case today. How about lack of oxygen? So oxygen deprivation, got it that time, is going to block the electron transport chain and shift us into anaerobic glycolysis. And so when we talked about the shift, especially in skeletal muscle, from anaerobic, aerobic, and back and forth, we talked about muscle oxygen deprivation. This is the mechanistic reason right here, is because your electron transport chain is getting stalled.